this is a nice segue into what Mace Rothenberg from Pfizer is going to talk to us about, because I think the critical piece here is we have crizotinib, which is where Alice has shown fantastic data on, and the next step is how does one move a biomarker-directed therapy after identifying the right biomarker for regulatory issues. So Mace Rothenberg from Pfizer.
Thank you, Nooper, and thank you to my former boss, Bruce Chabner, for inviting me, and to good friends, colleagues Marty Murphy and Dan Habron and Bob Pinedo, and so many of you here. So it's good to be here. When I wrote the title for this, I didn't intend, but sitting here, I realized that there's really a double entendre here. So what I intended initially was for dream to reality, taking something that was really aspirational and then reducing it to practice. But also, we know that when you're dreaming, things always look better than they do in reality. And so this is the harsh reality of trying to develop a compound in concert with a diagnostic. And these are the learning objectives. So clearly, there are multiple benefits to trying to target the patients who are most likely to respond to a therapy. First, we can design our trials to have a bigger treatment effect because we're selecting patients who are most likely to benefit. In addition, because we're looking for a larger treatment effect, we can actually detect that with a smaller number of patients than the typical unselected trials. From a commercial perspective, certainly something we all endeavored to do is to select out those individuals most likely to respond, and instead of seeing the kind of variability in length of treatment that we see, that in these selected patients, we'll have them on treatment for a longer period of time because they'll be responding. So it ends up having a development path that's less expensive and faster to completion. We achieve earlier regulatory submission and launches. We demonstrate dramatic treatment effects that facilitate early regulatory approval and demonstrate the robust value proposition to facilitate access. So with crizotinib, as you've heard, we really had a double whammy of accelerating drug development, but in a newly defined patient population. We were creating a new disease, if you will. So the opportunity was we saw unprecedented anti-tumor activity in refractory non-small cell lung cancer patients. It was active in a setting in which there was no standard of care beyond second-line treatment. And the anti-tumor effect was clinically meaningful, and the story that you heard from Alice in terms of patients who reported improvement in symptoms within weeks of starting treatment has been repeated and recounted to us on a number of occasions. And having been in clinical oncology for more than 20 years, I can tell you that's what often would save the day for me in clinic, to see just one patient like this who had such a dramatic response. And not only did it have a great impact on the patient, it had a great impact on me, the clinic staff, and others. But there are also challenges. So who are these patients, and how can we identify them? There are only 5% of all patients with lung cancer. How can we inform physicians, and why should they care? Is EML4-ALK prognostic or predictive? These are questions that we were facing early on. It could have been that we were just selecting a group of patients who had a better natural history, and that would have misinformed us to thinking it was a drug effect when it was really just selection of good prognosis patients. Fortunately, Dr. Shaw and colleagues addressed this in a publication last year in JCO, which demonstrated that there was really no evidence of this being just purely a prognostic factor in outcome of patients with this form of lung cancer. And then what tools could we use to identify these patients? As you heard, the initial detection and confirmation of EML4-ALK fusion gene status was done by FISH in a laboratory-developed test here at Mass General by Dr. Iafredi and colleagues. But this is not sufficient for broad application. I think Dr. Iafredi has other research interests and would be just overwhelmed by having thousands of samples descend upon his laboratory every year. So we really needed to address how we can scale this up and make this commercially available and appropriate. So this was the IUR, or the investigator use only, and leading to a PMA, pre-marketing approval approach. And then how do we balance ease of use and accessibility with the body of knowledge generated with one platform or test? I didn't say against a gold standard because we don't yet know what the best and most accurate assessment of ALK status is. We have the most experience with FISH, but we're also now gaining experience with RT-PCR and immunohistochemistry. So, well, we'll come back to that a little bit later, but you can just see that now as you get into some of the details, this becomes a real challenge. So this is the outline of my presentation. So is there any precedent for this? Well, from the approach of a sponsor, we could use gefitinib, which came on the scene about 7 years ago, when it was originally given accelerated approval based on the results of several phase 2 studies showing activity in patients with refractory lung cancer. Therefore, it had a very large market potential, more than 500,000 patients a year in the US and the EU. However, later on that year, the 2 phase 3 trials that were meant to confirm the activity of this drug in non-small cell lung cancer failed to show that gefitinib added to the benefit of chemotherapy. And therefore, because they're unable to confirm the preliminary activity seen in the phase 2 trials, the use was restricted, and basically the number of patients who could get this drug in the US and EU dropped to 0. 5 years later, now we first heard about the IPASS results from Tony Mock, in which patients who were selected based more on phenotype, patients who were more likely to have the lung cancers that had EGFR tyrosine kinase mutations were enrolled, showing just a remarkable improvement in progression-free survival, and using it in the first-line setting. So now this has rejuvenated the market in the US and the EU. And now, most recently, the genotypic subset, those individuals whose tumors really do have the EGFR tyrosine kinase mutation, have had even more dramatic effect. And now this population has been clearly identified and will likely lead to renewal of regulatory approval in the United States in the near future. What about the approach of the regulator to diagnostics? Here we could look at the EGFR antibodies. In December 2008, based on retrospective analyses demonstrating that those colorectal cancer tumors that harbor KRAS mutations were unlikely, in fact, at basically 0 response rate compared to those who had wild-type KRAS, whose response rate was 30%. This was rapidly identified and actually taken up by the EU to be able to help direct EGFR antibodies to those patients with colorectal cancer whose tumors were KRAS wild-type. Interestingly, in the United States, at ODAC, the FDA's position was that was retrospective. That was done with differing assays, and they felt uncomfortable using that to guide use of this drug. Well, having previously been in the GI cancer arena, I could tell you many of us shook our heads about this because we were thinking, given what we knew, that it would be unethical to randomize patients who we knew had KRAS mutant tumors to an EGFR antibody. So fortunately, that perspective prevailed, and about 6 months later, the FDA listened to the fact that this was an impossible task to do this trial and also unethical. So while they said they would prefer it to be done prospectively, they eventually added in late 2009 KRAS testing as a selection factor for patients to receive EGFR antibodies, and now that's a part of the label. So as you can see, this has been done, but not very well in the past. So how do we do better? And all of you are very familiar and very aware of how difficult it is to develop a cancer therapeutic. But to try and develop a cancer therapeutic in tandem with a cancer diagnostic really increases the challenge. We talk about it a lot, and certainly it is an aspirational goal. And the next few slides really step us through what the ideal process would look like. And these are a number of things that, being in the therapeutic part of things during my whole career, I really wasn't very aware of. So it really starts with discovery. Just like drug discovery, this would be target discovery. You identify the target, and immediately the question is a companion diagnostic for this? And what could be used? Certainly we see a lot of Western blots, a lot of sophisticated genetic analyses done from laboratories, but those are done under very controlled circumstances with basically unlimited access to the tumor. This is something that is not often the case in human samples, so we have to begin thinking very early on what is the relevance of the findings of the biomarker in the laboratory to the clinic. We can then use this to identify initial target patient population. We can use it to determine whether or not the drug is hitting the target, so a pathway readout, and then we have to determine how best to assay it. So the goal is to identify the biomarker very early. The next stage is biomarker validation. So how do you know that the change in the biomarker correlates with any meaningful output? So you may need to evaluate multiple technologies and platforms. Will this be applicable to just that tumor type in which it was studied in the laboratory or to other tumors as well? And again, I just want you to think of the EGFR story to realize that even though it's the same growth factor signaling pathway, its biological means of activation and resistance is very different whether we're talking about lung cancer or colorectal cancer. So it's not just all biomarkers, it's biomarkers in the particular setting of that organ. And then certainly simple is better, so we need to establish assay reliability early. The next stage is assay development and analytical validation. So here we prepare a formal strategy for selection and development of the biomarker. We have to decide whether or not what we know about this tool is enough to allow us right out of the chute in phase 1 testing to begin to select patients, or whether we need to collect all patients, unselected, collect the information from their tumor samples, and then be able to analyze those in a retrospective fashion. Certainly with the BRAF story and the development of sorafenib, if the feeling had been with such confidence that BRAF was relevant in melanoma, this is a BRAF inhibitor, the development pathway should only go to melanoma, that would have been a drug that never would have made it to market. But it was because of the broader testing in multiple tumor types that its inactivity in melanoma was appreciated, and its activity in renal cell cancer was appreciated, and then further understanding about this being also a VEGFR inhibitor. So here, we have to then be able to identify the IUO, or investigator use only companion diagnostic, that will then be able to bring forward into clinical testing. It occurs, and this requires validation of components, interaction with a diagnostic provider, and then really beginning to work out the logistics so that this is now ready for early clinical trials. Then comes clinical utility testing. So actually, is the diagnostic performing the way you thought it would perform in the clinic? In addition, here you begin to address some practical questions like, are all tumor samples created equal? If you have a surgical specimen where you get grams of tissue versus a biopsy where you get much less, milligrams of tissue, versus a fine-needle aspirate, what if you have fresh tissue versus fresh frozen versus paraffin embedded? What if you analyze the sample immediately after collection or after a certain period of time, and at what length of time? So it turns out that these are all questions that the agencies want answers to. So it's not all that easy. That's why when we are asked why can't crizotinib move forward even faster, we're busy addressing these questions as well. But I think one of the points to take home from this is that really is a dialogue between the agency and the diagnostic partner and the sponsor that really can make this a successful endeavor. And then lastly, regulatory review and approval. We need to, again, coordinate with our diagnostic partner in terms of timing and availability. We have to then understand what the requirements are not only in the United States, but in the European Union, Japan, China, rest of the world, and those requirements are different. And then begin working on other tools as well that might allow us to make this even more available and more accurate. So now, how do we select the assays? And looking at the crizotinib story as an example, really we looked at FISH, IHC, and PCR. So the advantages for using FISH was we knew it was highly specific and sensitive. It was applicable to all ALK fusions, and the technology was widely available in many academic pathology labs. As we've since learned, not all community laboratories have similar capabilities. But there are also some deficiencies. It was technically challenging. That slides required trained eyes to read. There was limited availability of this technology in Asia in some parts and South America. And it was not usable at the time with bronchial lavage or sputum. Well, what about IHC? This was widely available, could be done in community as well as academic medical centers. technically less challenging than FISH, and certainly a cheaper assay to run. But here early on there are false negatives and some positives. Sometimes further confirmation by FISH was required, and this is also not usable with bronchial lavage and sputum. But one thing that we've seen over the years that this has been an evolution is that technology will change and evolve. And some of these limitations are now no longer such significant limitations, and there are now studies being done to actually look at cross-validation across these platforms. And lastly, PCR. Here, the advantage is a possible multiplexing of assays, which I'll get to later. Again, widely available, and this also could be applied to biofluids and circulating tumor cells. But here, with PCR, you have to be ready to test all the known variations and then be open to the fact there will be some unknown variations. The ability to extract this from FFPE samples may be challenging, and then, again, limited to known variants. So when all is said and done, when we looked at this, the successful use of a FISH assay here at Mass General in Phase 1 was an important consideration for the choice of the assay going forward into the pivotal trials. Now, another issue that we all face is the availability of tissue for testing. And as you all are acutely aware of, these assays have not been developed in any coordinated fashion. So to test for EGFR amplification, we use IHC, EGFR mutation, PCR, EML4-ALK, FISH, and you can read the rest there. Most patients who have lung cancer diagnosed by a biopsy have very limited tissue available. So how do you decide which of these to do? And what technology is going to give you the most information? So this is one of the big challenges in the diagnostics world, and for all of us, is to put all these on a single platform, so you can actually look at the most relevant genes and mutations in a single sample on a single platform. That will make it easier to do with a high percentage of samples. It will also bring down cost and make it more widely available. So, challenge of choosing the right diagnostic partner. It's, as you've heard Bill Sellers mention about trying to coordinate the development of drugs across sponsors. It's also challenging to coordinate your drug development with the development of a diagnostic that's out of your control, but yet this was absolutely necessary. Most companies that develop drugs don't have expertise in diagnostics, and therefore we needed to develop a close collaboration with a partner. Fortunately, we have with Abbott Molecular Diagnostics. We recognize that pharma and diagnostic companies have different set of expectations, cultures, and reimbursement paths. We have to perform due diligence on the pros and cons of each technology platform. The selection of a diagnostics partner should occur early as possible, and then close collaboration with diagnostics companies necessary for IP ownership, assay development, manufacturing, and the rest. Challenges with assay development. One of the small details that we realized was originally Abbott wanted to do FISH testing in centralized laboratories, which seemed very efficient. They would have one facility in the US, one in Europe, one in Asia. They would have the most experience with the test, and you'd have that experience ensuring the similar kind of quality across these. Well, it turns out many centers don't like sending their precious samples outside. And especially when they have capabilities of doing those tests internally. In addition, there are some regional laws we had to be aware of. So, for instance, in China, law does not allow any biologic samples to be sent outside the country for analysis. So we had to address this and work with the sponsor to be able to roll out a kit that could then be done at the individual sites, and that's really allowed us to enroll patients much more briskly than we would have otherwise. I told you a little bit about the sensitivity and specificity, taking into account the limited number of patients. So when we engage with the part of the FDA that focuses on in vitro diagnostics, the Center for Devices and Radiologic Health, CDRH, they're used to reviewing and approving new tests for testing LDH levels or hemoglobin that you could then look to validate and determine the accuracy on thousands of samples and come in within the gold standard by a very tight percentage. If we needed to test 1,000 patients with ALK-positive non-small cell lung cancer, we'd be looking at not having this drug around for 10 years. So it really has been educating that part of the FDA along and working with the Office of Oncology Drug Products for them to take another look about what criteria are necessary for the development of a diagnostic for cancer patients and how they need to be flexible on that. And although you'd like to think that there are regulations that guide us on this, there really are no regulations that are specific enough to handle this. So we're really finding our way together, but it has been a good and very productive collaboration. So working with regulatory agencies to look at these issues, to gain experience with as many types of clinical specimens as much as possible, and establish clear guidelines for specimen handling are important. So the sponsor's perspective. When this first emerged as an opportunity, remember this is a drug that we brought into clinical testing because of its activity against MET. In fact, we brought forward a backup compound because we thought the ALK effect was off target and the backup compound was a pure MET inhibitor. So it just shows you sometimes it's better to be lucky than good. And so here we were facing this wonderful dilemma of having dramatic responses but in a small group of patients. So even when you add together not only the non-small cell lung cancers, but the anaplastic lymphomas, neuroblastomas, and sarcomas, total is still less than 10,000 patients. Fortunately, and in industry, again, one of the things I've learned is you have to work within a larger team that may not be driven solely by the scientific attractiveness of the opportunity or even the clinical impact in a small number of patients. You really have to be able to make it a commercial success. So fortunately, I have a wonderful partner, Allison Ayers, who heads our commercial development group, which has more than 20 years of oncology experience. And when she saw these numbers, she was undaunted. She said, we can make this work because if it works as well as we think it does, these patients will be on therapy for a long period of time. So that was very nice to have that ally working with me. Then reimbursement and coverage. Who should pay for the test? Should it be patients? Should it be insurance companies? Should it be the sponsor? Who should be tested? Every lung cancer patient? One of the interesting things, if you read the original mono paper from August of 2007, the cell line in which the original ALK fusion gene was detected in was derived from a 73-year-old lifelong smoker with squamous cell cancer. So, who should be tested? How should the diagnostic be priced to make it accessible and not a barrier? The regulatory approval of diagnostic is not a guarantee of reimbursement coverage, and therefore studies are underway right now to understand the health economic outcomes and understand that may be varying paths of coverage to cost of diagnostic around the globe. And then when we look to the future, what is the ideal stakeholder engagement? What will it look like? Well, certainly pharma wants to advance personalized medicine in the development paradigms with integrated biomarkers and companion diagnostic development. For a diagnostic company, it's to be integrated into that process and be able to detect these changes with minimally invasive technologies and multiplex assays. For physicians, as much as all of us in this room speak pretty much the same language, when you go out into Tulsa, Oklahoma, or Springfield, Tennessee, you're not going to find many people in practice who understand molecular biology very well. So there's real education that has to occur for people to understand the benefit of these new therapies, and a commitment to molecularly guided treatment decisions. For payers to recognize the overall value proposition of personalized medicine and to ensure access and value-based pricing for treatment and companion diagnostics. For regulators to streamline the regulatory approval process, and for patients to demand molecular diagnostics and personalized treatments. And lastly, it's adoption into practice. So you've seen this slide in various forms over the years where we've basically gone from two main categories of non-small cell lung cancer, adenocarcinoma or squamous cell carcinoma, based on whether patients are eligible to receive Avastin with their chemotherapy, to the future paradigm to be able to divide this into now 7, probably more categories in the future, and then to be able to use that information to direct the patient's treatment. One person that we haven't mentioned is the pathologist and how critically important the pathologist will be in the success of this approach. We need to engage them so that they know about the appropriate storage and timely retrieval of tissue, the identification and ordering of the right molecular diagnostics test, turnaround time to support treatment decisions, interpretation of results and subsequent treatment choice, planning tissue for multiple tests, and sequencing and prioritization of tests. Without their buy-in, and in fact, without the transformation of our clinic laboratory ordering sheets to be able to just check off one box as you do for a CBC, to check off one box for profiling, molecular profiling for lung cancer, is going to be a critical barrier for us to overcome in order to make this easily accessible. If it's tough, physicians from practice won't do it. And then what are the barriers? Some of you may be surprised that patients may be a barrier to the adoption of this because it may involve some treatment delays. And then multiple procedures to obtain tissue may be considered unacceptable to patients unless we educate them about what this means for their care. Physicians may be the burden of education to incorporate molecular diagnostics into treatment decisions and the logistical complexities of processing the tissue and then interpreting the results. And then the cost, mentioned earlier about multiple unique platforms and multiple potential tests for each patient and inefficiencies of the system. So the lessons learned with crizotinib, the rapid development of crizotinib was a result of a combination of good science, good communication, good collaboration, and good luck. The observation made came just at the right time in the drug's development for us to be able to quickly move and shift the focus into ALK fusion gene positive non-small cell lung cancer patients. Early realization that companion diagnostic would be essential to the development of crizotinib. We couldn't develop a drug that only benefited 5% of the patients in an unselected group of lung cancer patients because that effect would be so diluted as to lead to a negative outcome. The importance of early and frequent interactions with the FDA in shaping the clinical development plan for crizotinib, and the ideal scenario for co-development of diagnostic and therapeutic will rarely occur. It certainly wasn't ideal for crizotinib, but as we think about what that would look like, we have to recognize that the selection of a diagnostic early in the clinical development may lead to there being some dissatisfaction with how primitive that is in the years during which the drug is under development because technology moves forward. We may not have complete knowledge of the target or understanding of the target's clinical relevance. And so lastly, what we can say is that the signal of clinical efficacy in a select group of patients is the beginning, not the end of clinical drug development. The principle of precision or personalized medicine is obvious. Its reduction into practice is a little bit more difficult and requires careful consideration of those factors listed: scientific, clinical, temporal, practical, regulatory, logistical, ethical, financial, and collaborative. But despite these challenges, the benefits of personalized medicine to patients and to society make these additional efforts eminently worthwhile. And I'd like to last thank my colleagues. This really was a multidisciplinary collaboration and continues to be so at Pfizer. In translational oncology, Akan Sakul is charged with overseeing our diagnostics and biomarkers group within Richard Buller's organization. In regulatory strategy, Erling Donnelly is the lead regulatory specialist for crizotinib, overseen by Ramsey Dagher, and Allison Ayers, who heads commercial development. Thank you very much for your attention.
Thank you, Mace. That was terrific. We have time for maybe one or two short questions.
That was a great talk. And one of the things that I think a lot of us in the industry are struggling with and that you didn't directly address, so I'd like to ask is, to what extent do you need exclusivity and an intellectual property position in order to make it reasonable to commercialize a companion diagnostic in the context of a pharmaceutical company?
You know, that's interesting because as far as my understanding of the agreement is, there is no exclusivity of this. They have, the Abbott Molecular Diagnostics will have the experience of using its diagnostic in the clinical trials and being able to correlate results with outcomes. But that is not exclusive, so other platforms will come along and then can be able to address some of the potential shortcomings. So it's certainly in Abbott's interest to be able to make this FISH diagnostic as attractive and easily accessible as possible.
Yes, I was wondering if you could comment on essentially the ethical complexities of designing late-stage clinical trials with a drug that you know will only, when you've sub-selected patient population that you feel will likely work, and then have to put drug or deprive the patients of that drug and to do a randomized trial?
Good question, and that's why we're not doing only the randomized trial. We're doing the single-arm trial as well, so that patients who are randomized to the chemotherapy control arm of the phase 3 who do progress can then receive crizotinib later on. But also, we were very careful to design the trial with 2 drugs that are FDA approved for second-line treatment of non-small cell lung cancer because of their perceived efficacy in that setting. So the feeling is that there has to be a certain amount of clinical equipoise because there are going to be patients who benefit from those drugs. So, and we did not know, certainly with the emerging data, we're becoming more confident about the robustness of these responses. When these trials were designed, we did not know how long these would last and how robust they would be. So we were trying to tread a middle ground here to be able to perform trials that were going to be able to establish the benefit of this drug in patients with non-small cell lung cancer. Initially, this drug is likely to be reviewed on intermediate endpoints like progression-free survival. And if that happens and the drug gets accelerated approval, let's say, or Subpart H approval, there is a regulatory requirement that we perform now more than 1 phase 3 trials in order to confirm the effectiveness of that in a randomized controlled setting. So it really is a dialogue that we have with regulators to be able to work together towards reaching the kind of clarity of effect that we really seek so that when these trials are done, we know what impact the drug has, not just on intermediate but ultimate endpoints as well.